Okay, so we're going to do option B, which in the old days was option C. That's the purpose. That's why at the top of this screen, which is not scrolling, it says option C. I'm also going to open the Chem Guide, make sure we see the appropriate regions. Check the website for that, so I'll be going through these very briefly right now. And also the data book has some important compounds, so let me scroll to some of those. Okay, so the first thing you're required to know, here is the first point. Now, you guys already know this. How do you calculate the energy content of food? You burn it, and you determine, I know it sounds funny, we burn it, and we determine how much heat is absorbed by water. That's it. So remember, the heat absorbed by water, Cliff, remember your formulas? Cliff said there was no formula. There's the formula, specific heat capacity times mass times delta T. This is the heat change, so this would actually be absorbed. So basically you burn it, you'll detect a temperature increase in water, you use this to calculate the energy change in the water, which you change the sign of and becomes the energy lost by the food. Right? Remember that? Yeah? I'll take that as a yes. Okay, so going back here. Here is an example of it. I know it's small. Let me see if I can change the zoom level here. Oh, it's going to have to do it down here again, huh? Okay, so here's an example. Right here. Now, notice 37.5 grams of breakfast cereal gives 540 kilojoules of energy. So there's the Q. Notice, we plugged it in. We calculated, it says calculate the temperature rise in a food calorimeter with water of 625 if 1 gram of the cereal is burned completely. Okay, so 1 gram of cereal, here's the ratio between grams and kilojoules right there. So this is going to convert grams to kilojoules of heat. That's Q. So we're going to solve for delta T. That's all we did here. Q, mass, specific heat, delta T. Does everyone understand how to calculate it? It's basic thermochem. Any questions there? Okay, I'll take that as a no. Okay, next one. Proteins. Okay, let's slow down. Go proteins. We love proteins, except when we have to do something with proteins. Okay, so proteins, the subunit of a protein is an amino acid. Very important lesson here. This is an amino acid. This is a generic amino acid. I put a subgroup of R there because in that spot can be different chemical groups or different, could be carbons, could be other things. There are some sulfurs, there are all kinds of things that could go down here. And your chem book, there they are. Let me make sure this is the right form for you. That's HNMR data. Here they go. So here are all the amino acids. You'll notice here, for example, there's a CH3 group. That's alanine. Look at that big old mess attached in the R. The only difference in all of these pictures is the attached R group. The backbone is identical. This portion is always identical. Now, this drawing you see here is never, ever, it's never going to look like this. Amino acids will never look like this. Okay? The reason for this is, if you look, you should recognize that. It's a carboxylic acid group. This is an amine group which has basic characteristics. This compound can serve as both an acid and a base. So therefore, it has function as a buffer, which you see right there in that bullet point. So you have to understand how it works as a buffer. Okay, now let's be very clear about this. On a pH scale, this is very important. It's very important for proteins. Very, 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 very important. Okay? Now, when pH is below 7, that means we have a lot of H+. Agreed? And the pH, it's acidic. Agreed? So, imagine this chemical. Forget the R. It's irrelevant for right now. Just focus on the backbone. We are in the presence of a lot of H+. That means that this will be present because that's an acidic proton. It can come off to form a negatively charged ion, but because we have a lot of H's, think of this as Le Chatelier's principle. We are overloading this compound with H's, so this is protonated. This is a base. What does a base do in the presence of acid? It accepts them. So the form present, the form of an amino acid present below a pH, at low pHs, I should not say exactly at 7, but at very low pHs is this form. Notice the extra H. Now, I'm not going to be able to show this very neatly on my drawing because I suck at art. Okay? But this is very important. Now imagine the pH starts to climb. So we start to remove H pluses from solution. Now look, there are two sources of hydrogens to come off first. You with me so far? There are two sources to come off first. This one would come off first. So let me write a little note to us first. So which one of these two? I take that back. I misspoke. So here's our low pH form. As we start to increase the pH, meaning we're removing H pluses, okay, the acidic proton right here loses it first. So here's what you have when that acidic proton comes off. Here's that NH3 group on the end with its net positive charge. There is the carbon in the center with whatever R group we happen to be talking about. And over here, we've lost that H. We have a negative charge. Now notice, we have one compound with both positive and negative charges present. This compound is neutral. There is no net charge on this compound. This compound, at low pH, has a net positive charge. If we continue to increase the pH, meaning removing H pluses, or you can even think of that as adding OHs. What is the next part of this that's going to lose the H? Over here. So at extremely high pH, here's what you have. What's the net charge on this one? Now notice, high pH, net negative charge. Low pH, net positive charge. In the middle, neutral. You understand? The pH at which the amino acid is neutral is known as PI, isoelectric point. At that pH, the amino acid has no net charge. It is neutral. That is very important for separation of proteins. Now, if we go to our data book, look at this chart. Here are those amino acids. Notice this column here, pH of isoelectric point, PI. Okay? So in other words, at a pH of 6, alanine is neutral. Below 6, negative. Below 6, positive. Above 6, negative. 10.8, arginine is neutral. Below that, positive. Above that, negative. Neutral at 5.4. Below that, positive. Above that, negative. Do you understand? So that is a technique for separating protons. It's the basis for a technique for separating proteins. So if I go back to this chart, let's look at what you're required to know. Draw the general formula. There it is. That's that. Understand that there's a backbone and a unique R group. The properties are the isoelectric point, the formation of a Zwitter ion. I don't know. I used to know where that word came from. It does say Zwitter ion. That's this issue we just discussed. A Zwitter ion is this one in the center. Okay, it's the fact that it changes in charge as it goes up and down pH.
Wait, so the Zwitter ion and the isoelectric point are the same?
The Zwitter ion is what it is. The isoelectric point is a pH. The Zwitter ion is the name we give to that ion at that point. And the fact that it's a buffer, meaning it can both absorb and release hydrogens. Those are all reflections of the same exact issue. You're supposed to describe the condensation reaction. What does a condensation reaction produce, by the way? This is biochem. What do all condensation reactions in? Water. So I'll use a generic one here. Notice I'm drawing the basic 2 amino acid. It's called 2 because this is carbon number 1, this is carbon number 2. That's why they call it 2-amino acid. I'm not drawing any ionic forms of it. It will connect to a neighboring compound. Here's another amino acid. One of these H's and that will go away. And these two will connect to each other. That is a condensation between amino acids to form a protein. You've probably done that in biology. This is called a peptide bond. This is all, dehydration synthesis. If you were to separate them, it'd be hydrolysis, water splitting. Okay? Basically, you put water back in. Okay? Pretty basic stuff. Describe and explain. Oh, this is going to be taking me a second or two. Describe and explain the primary, secondary, tertiary, and quaternary structure of proteins. Watch how good I am at this. Watch how good I am. Yeah, for you folks in advanced bio, it'll be a little simple, a little more easy. So, okay, primary. It's the sequence of amino acids. So look, I know it's tough to see the mouse cursor, but here is one amino acid right here. There's the peptide bond. Here's a second amino acid, another peptide bond, another amino acid. This is 3 of them in a row. You're required to know up to 3. Notice that they still use the R groups. Remember, those could be different R groups. That'll play a role in a second. That's the primary structure, a straight line. These R groups and these other parts of the compound can interact. So these chemicals are sticking off in all different directions. They can attract each other. They can repel each other. That is the secondary structure. The 2 most common ones are called alpha helix. This little curly string is the primary. These dots are supposed to represent side groups. They can interact and cause twisting. That's an alpha helix. This is called a beta sheet, sometimes called a beta pleated sheet. All you have to understand is that they are secondary structure. They come from side groups interacting, repelling and or attracting. Okay? Now, those secondary structures, which are within a strand, those secondary structures can cause different areas on the same string of amino acids to interact. So imagine that this top row is one strand with its interactions, its secondary structure here, and here's a here is the same strand on the bottom far away. So you could imagine that this string of amino acids continues off in this direction and loops back. So this strand acting with itself further down the line, that is tertiary structure. Think of it as a spaghetti strand that is interacting at both ends. It's looping around and both ends end up sticking next to each other. That's tertiary structure within one strand of amino acids. Does that make sense? And quaternary, which means 4th degree, is where you have different spaghetti noodles coming together. Well, I mean, look at it. It looks like this diagram looks somewhat like spaghetti noodle. This portion would be the strand. Notice in some parts it's looped back, so maybe right here you could say there's some secondary interactions here. But further down the line it interacts with itself even more, which would cause a tertiary structure, 3rd level of structure, and then those